lesson now in the University of Texas University, MD Anderson. So, and my field mostly focused on blood cancer. And so when I was doing my PhD, I focused on colon cancer cell, and now I focus on CAR T cell therapy that to treat patients who are left with, multiple treatment like chemotherapy or radiotherapy. So, um, and I did my PhD in France, and so after that I came back to USTS, and I worked there for nearly years, and after that I go to a state to do my postdoc. And now I'm in Houston, Texas, and so today, so we go to genome edit podcast. So for the general of okay, the lecture today we have like so actually Dr. Ming also go to like two lecture right now, right? Okay, and so now we actually so we see we'll come back to lecture 3. For next week or the following week. So first of all, before going to the lesson today, I would like to know if there any student that did not study at USTS in their graduates, undergrad, like in bachelor study. Any student that anyone? Me. Okay, so can you guys introduce a little bit about yourself first, like about your knowledge, about background, so I will have some idea about it, and I will go to the lecture, and maybe you, right? Mm-hmm. Oh. Mm-hmm. Okay. In general, so did you have any idea about gene technology, any kind of gene technology that you know? Hmm. Okay. Uh, Anyone else? I think we have at least 3, right? The second one, second.
Yes, I graduated in Hanoi International University of Education.
Mm-hmm.
Yes.
Okay. Do you have any like background about genetics, about genome editing, about biotechnology?
In my bachelor course, I have learned about biotechnology, but because I have forgot. Yes.
Okay. Okay. So you know in general about some techniques, right? Like some basic, right? Basic techniques.
I have learned the basic knowledge, not detailed.
Mm-hmm. Okay. Anyone? I mean, anyone else?
Yes, I
Mm-hmm.
graduate from Vietnam National University of Agriculture.
Mm-hmm.
Uh,
Okay.
and my major in the university is not about biotechnology too.
Okay. 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 Anyone else?
Yes, I graduated at Hanoi University of Science, and my university major is biotechnology.
Okay. Okay. Yeah. And next one.
Stage.
Anyone else?
Sorry, my cam work not good. I'm not having the
Yes.
phone.
Okay. So. Mm-hmm.
But I was graduated from Hanoi University of Pharmacy, so
Right.
we don't have the background much about the genome editing.
Right. 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 Anyone's? Okay. Okay. Let me 1 minute because I want to fix all the webcam. So I want to see all you guys. So. Okay. So now we'll talk a little bit about CRISPR-Cas. Um, so the lecture today we have some like main parts. Like we will go quickly about introduction. I don't think I could talk too much about introduction, and then we go to the CRISPR-Cas system, and then we go to the CRISPR-Cas in bacterial immune system, and then we move to engineering and application. And we have experimental design, but I don't know if we have time for this, but I will try my best to finish soon and to reduce the time to projects. So for the technique, so basically the DNA editing is the technique that based on the working of the enzyme that's bi-specific to the DNA, and then it can cut the DNA. Cut the specific sequence based on the guide RNA, and then so the the cell can repair. So in the cell we have like some machinery. I mean enzyme and to repair the cut. And for the purpose of CRISPR-Cas, so we can use this technique for target gene mutation, and we can use it CRISPR-Cas for gene knockout, and also to study gene function. For example, to study gene expression, and also to use the system for to locate some gene sequence and to know the expression of some gene. And actually, for CRISPR-Cas, so firstly, normally in the past we have some like different system that can cut, can break the DNA sequence. So normally in last few decades we can use meganuclease, and we can use zinc finger nuclease, and we can use talent and also CRISPR-Cas9. So CRISPR-Cas9 is just it's studied like few decades ago, like in 2019, but it's just become popular like last 2 decades. So did you anyone know about this system about like CRISPR-Cas or know about the gene finger nuclease or anything in your bachelor study? Any ideas? No. Okay. Did you guys know about like oh sorry like how in how the enzyme, how the bacteria prevent or protect themselves from virus infection or anything? No idea. Okay, so firstly, so we have the enzyme. So all this enzyme can, I mean, works the DNA a specific reason, specific sequence, and then it lead to when the DNA breaks, so it lead to the, I mean, the cell have a mechanism. So I mean, they have the whole system that can fix the DNA breaks. For example, in this case, so when double DNA works, so some enzyme in the body, I mean not the body but in the cell, can go to repair this one, can repair it. So because we know about this one, and yeah we use this technique to design, I mean to knock down the cell and to knock down the gene, and also to um, uh, focus on some gene technology. So actually we have for for cell repair we have two mechanisms to repair the cell. So we have two ways. So firstly we have a non-homologous and zoning repair. So it means that so when uh, this when this DNA break down, so it makes the overhang, it makes an overhang, and after that the enzyme can, I mean this oh maybe I will search if I will search an example for so this one. So for example for oh sorry, so this is a mechanism for non-homologous and during repair. So for example, when the enzyme cuts the DNA, so it makes an overhang like this. So it makes overhang. So the two overhang, and then in the body we have different enzymes, so it can come to repair this one, and then it's joined to different strand. I mean to different to join two different fragments of the DNA. Yeah, they join together. And And for second mechanism, so when the DNA, when DNA is break down by enzyme, so and then it makes like it's not how to say, it makes uh it not makes overhang. It doesn't make an overhang. So and then so the enzyme can join to the DNA directly. I mean to this one. So we can say that a hormone direct repair. So sorry. Um, do I understand about it? Two different methods, two different mechanism, two different way. Sorry.
Can you explain it again?
Okay. I'm so sorry because so this is my first time give you the lecture, so I'm a little bit so like unconfident. So okay. So firstly, we have two different. So actually, we have two different mechanism for repair. So firstly, when the DNA is break down by enzyme, so we have different enzymes that I mentioned before. So when is we have two mechanism in the cell to repair the double strand breakdown. So for example, in this case is equal non-homologous and joining. It means that when the parental DNA breakdown, it not breakdown like complementary, like you have blunt overhang like this. You don't have overhang. So for example, here you don't have an overhang. You have like sorry. So the one you don't have an overhang. So the one you don't have an overhang. So it can go to the direct repair or direct comparison. And in this case, it makes an overhang. So we can see you in this position you have no complementary. We have no null, right? So it missing some null here. It missing some null here. So it means that the cell go to the cell go into the non-complementary repair enzyme. So firstly, it need to add some nucleotide into this overhang and to make the complementary. Yeah, it make by some enzyme like 1.3 in the cell, and then it go to it like it's like it two different fragments. This one it like it normally it can like it by like yeah inside the cell we have different mechanism. And so for for direct repair, when the cell, when the cell, no, when the enzyme breaks the DNA and it make an overhang, no, it makes the blunt DNA, two different blunt DNA like this. So we don't have any overhang like here, right? So, and then it go to the homologous, direct, direct repair. Is that clear for you?
It means that which mechanism of of repair, it depends on how the break looks like. If there's
Yes,
an
right,
overhang, then it will
right,
use the first one, and if there isn't, then it will use the homologous direct repair.
right, right. Because so when the enzyme break down the DNA strands, so it's, I mean, the true mechanism and the true pathway, the true way is occur randomly. It's normally it occur randomly, and in some case it makes overhang, and some case it makes just blind DNA. So if we have blind DNA, so it can go to direct direct repair, and if you have I mean overhang DNA, so it can go to non-homologous enzyme repair. So firstly, as I mentioned, so for example here, when you have like an overhang, so you need enzyme, some enzyme, for example, pole 1 on pole 3, going to that, I mean, that side, that region, to add, I mean, to add nucleotide and to make the complementary DNA. And then, so you can have an overhang like this case, like this. And then, so when you have two overhang strands, so it can join into, I mean, it can ligate two different strands. I mean, two breakdown strands together. So that are two different ways. Okay. So so based on this two mechanism, we have different. I mean, we have different approach uh, for using CRISPR-Cas9. For example, when um, so for example in this case, so in this case when we have like double strand, I mean breakdown, so the enzyme can insert, I mean in the body we have some enzyme that I mentioned before. It can join or it can insert some fidenase sequence, or it can insert some nucleotide into the breakdown point, or it can then let the whole, I mean the whole sequence, for example in this region. So here we have like indel mutation. And homo homologous direct repair, so in some case, as I mentioned, when we have I mean blunt DNA, so we can use this technique for introduce or for editing some gene sequence. Like for example, we can introduce, we have some I mean we have some DNA, and we can we can edit this one by replace some sequence, or we can insert some sequence, but um, we just repair, for example, we just create some mutation point in this region, but keep the whole sequence remain unchanged. So so I will not go detail about the history. So so this is how CRISPR-Cas9 works. So normally in the so in sequence we have different, like we have spacer and we have short repeat. So this spacer is encoded for, I mean for guiana. Later I will talk more detail about guiana. So this is the overall, I mean the structure of CRISPR system in the bacteria. So actually, as I mentioned before, CRISPR system is for like uh, it was discovered, it was studied in 1980, like few decades ago, but until 2020, I mean 2002, when um, Zanesen and his colleague have some, I mean, have some discover or some uh, study about the repeat DNA in prokaryotes, and then re- they they suppose that we can use that system for gene editing, and in until 2012, so Jennifer and Emmanuel were firstly proposed and firstly published research in um, um, I mean they was published a research and they proposed that using CRISPR Cas9 system for gene editing, and then in 2014 we have study that public fan, I mean by a Chinese scientist who using CRISPR-Cas9 to edit human genome. So they want to edit like um, a gene called CCR5, and so they want they hope that that so that edition also lead to HIV um, HIV resistance. So and this study were like have many like controversy, and it's also related to human right, and so that's why we still concern a lot about uh CRISPR-Cas technology in in human genome editing. Hi everyone, are you guys still here? Is there anyone here?
Yes.
Yes.
Still hear you.
Oh, I'm so sorry, but so I think we can like uh, because I still have no idea if you guys in chat or not, because I cannot see, and I don't know if my talking is mean to you, or that because like I just talk for myself, so it I don't know. Can you
Em bật nghe ạ.
yeah? Can you guys turn on webcam and so I can see? I can understand if my talking is fine or not, or if sometimes I need to slow down, or I need to speed up, or I need to change anything. Mm. Okay. So so this is how CRISPR-Cas9 work in bacteria. So actually in bat so actually first of all in nature bacteria can infect by different kind of virus. So normally the most know is like bacteriophage. So so when bacteria infected by some virus, for example, in this case, when the virus infect into the cell and it introduce, I mean, it injects the genome into the bacteria, and then in bacteria we have the CRISPR system. So we have Cas1 and Cas2. So Cas1 and Cas2 basically that's some nuclease, so it can digest bacteria DNA, and it make to it generates some fragment. Yeah, and then the fragment, I mean the viral DNA can insert into the genome of the bacteria. When it inserts to the genome of the bacteria, and then we have both space, like we have spacer, west spacer, and we have reason. So spacer that come from the, I mean, it come from viral DNA, and then the CRISPR-Cas system from the bacteria. Later, it can transcript it transcript the trace RNA and also the uh, CRISPR RNA. So the one we insert into the CRISPR RNA, and from this CRISPR RNA we use for, I mean, for future breakdown when uh, the cell reinfect by different kind, I mean the same kind of virus, for example. So here first of all I would like to say like bacteriophage, for example. For the first time when it infect into the bacteria into the cell, and so the cell break down the DNA and it insert the DNA into their own genome, right? And then after that, um, it has their own genome, and it also have the guy. I mean this DNA will transcript. I mean will make the guy RNA. Do you understand this point? I mean. Trần Xuân Anh, do you understand?
Khi yêu là không nói dối, hay là không nói dối như
Anyone understand this point? No. Phương Anh.
Like, can you repeat the part when it integrated into the bacterial genome? Because it's kind of confused in that part.
Okay. So it's still don't clearly about it, right? About it one, right? Tuyết Mai, do you understand?
Yes, I understand. Yeah, because my project now is related to CRISPR-Cas9. So.
Okay. Can you explain a little bit? Can you?
Yes. We can see that we have two parts in this system, immune system. The
Right.
first part is immunization, and the second part is immunity. And actually, I learned with three part. As the first step is adaptation, the
Right.
second step is expression, and the third is final is interference.
Mm-hmm.
So you can see that we have the viral DNA, and the viral DNA is trained the viral DNA is trained with the
Mm-hmm.
bacterial immunes. So we have the acquired sequence, and this integrates into the CRISPR arrays of these bacteria. Uh, this is the maybe this is the template for RNA sequence. We have the cracked uh, pre CRISPR RNA. We have the
Right.
yeah
Right.
the viral DNA of virus is CRISPR RNA, and in bacteria we have the track we have track uh, RNA, and we have Cas complex, and
Mm-hmm.
this complex is can bind with the CRISPR RNA,
Right.
and and build the CRISPR Cas the CRISPR Cas constructions, and
Right.
these structures maybe and now this CRISPR RNA is have the similar structure with the viral DNA,
Right.
and
Right.
and
Right.
this complex can cut the viral DNA to cleavage the viral DNA, and yeah, they can break this DNA.
Right. So actually, so the Cas complex is not only Cas9, but it have a different Cas. For example, we have Cat1 and Cat2. So Cat1 and Cat2, that's very, I mean, that is general, that very basic nuclease. So it's common to, I mean, it have in all system, like in bacteria, in cell, and different cell. So it's not so special for the Cat1 and Cat9, Cat1 and Cat2. So the Cat1 and Cat2 can break down and cut down DNA from virus. I mean, yeah. And after that, we have small fragment of the virus DNA, and so that amino DNA will integrate into, I mean, insert into the bacteria, I mean, bacteria genome. And after that, so yeah, you're right. After that, so this complex can express trace RNA. So this was the only trace RNA. And so here's the pole one, I mean, RNA pole three. RNA pole three will transcript all of this one. So actually, this spacer is come from different infection. It's not from the same infection, and so the bacteria accumulate the insert by, I mean, over their lifetime, like different infection, and they have different, I mean, different spacer. But so in the case we don't have example about this spacer, specific spacer, and after transcription we have CRISPR guide RNA, and we have CRISPR protein, and we have tracer RNA, and it all combine into the cat complex. Yeah, and this can recognize the DNA by I mean, it can recognize the specific um, uh, gene sequence, yeah, by this guy RNA. And then for the next infection, so because the bacteria have the guy RNA that can recognize the virus, so therefore it can break down the virus, and so it can, I mean, yeah, it can survive from infections. So actually we already know about the about the mechanism how bacteria can prevent delay from infection. So actually, as I mentioned before, we have different spacer. So if different pressure, it comes from different infection. So because the CRISPR-Cas, I mean, so the CRISPR-Cas needs RNA. So RNA is, as mentioned before, we have trace RNA. The tracer RNA. Tracer is trans-activating CRISPR RNA. So this tracer RNA need to bind to the CRISPR RNA. It binds to CRISPR RNA, and it have some repeat region and also have some hairpin region. So that is the domain that have the CRISPR-Cas9 can bind to the RNA. Yeah. So we have like in trace RNA we have some constant. We have some constant region that can bind to the trace RNA. The trace RNA is like normally is like universal RNA. So it's like it's sorry. Do we understand this point? Hà Nhung. Hà Nhung. Yeah, do you understand this? Do like do do you have any idea about do you have any idea? So I do know how the bacteria have different spacer. Why the bacteria have different spacer and how? So actually in so no. So we go back to this. So here we have different spacer. So for each ball, this we have one spacer. For example, this we have one spacer 1, 2, 3, 4, 5, 6, for example, whatsoever. But actually in bacteria, so we have like hundreds, thousand different spacer. Yeah. So how and why they need that much? Any ideas? Anyone else?
I don't
Yeah.
know what a spacer is.
I think spacer is from the forage. From the forage DNA of a forage genome from other species. So it's called spacer.
Yeah, but how can the bacteria get that spacer?
I think it's get from other organism. But I didn't remember clearly, but I had read somewhere that some of our genome we take the gene from foreign organism, and so it's helpful for the survival of the organism. So we keep that gene in our genome. Maybe it's the reason why.
Yeah, it's right, but So can you guess where does it come from?
It comes from the virus.
Right. Right. So but why we have different spacer? I mean, so one are different from the other.
Because it's the vector acquire new spacer from new infection from various virus.
Right. So I think when I have a good background about this, so
But
yeah.
actually, I listened
Right.
from this beginning to now, so I know that you have speak about that. I just repeat that.
Mm, okay. So so as we discuss about different infection, and so also Vân Anh, right? Yeah.
Yes.
So Vân Anh also mentioned that that's I mean spacer come from different acquired system, because when the bacteria infected by different virus, and every time they insert the viral genome into their own genome. So and for the new infection for the next time, so the virus, the bacteria know, the bacteria can recognize that same virus species, same virus strain, so it can break down the virus based on the DNA that they acquire, that they insert into their own genome. And so here, after like during their lifetime, and they acquire many different infection, and so they can pass this this genetic into the new generation. So therefore, for over time, like up to like 100 year, million year of devolution, so they acquire quite a big, I mean, um, they acquire quite a big um, foreign genome. Foreign DNA from different type virus. So that's the way how the bacteria can protect themselves. It can say like the nature immune system.
I love it two-way thing, but I do understand the mechanism of CRISPR-Cas9.
Right, right. Can I talk a little bit about week one?
What?
So
Can you speak louder?
can you speak in two different way? Like in week way, it go to non-homologous and join repair, and in weak way it go to like homologous repair, direct repair.
From the lecturer, as I know, then this really depends on what is if it's the blunt end or it's the hanging end. So if the hanging end is gonna be non-homologous way, on if it's blunt end, then they will be the homologous way.
Yeah. So basically, okay. Phương Anh, I think Phương Anh very good about this. I know Vân Anh very good. Phương Anh. So Phương Anh.
Yes.
Yeah. Do you know how So how the bacteria can immune from virus infection?
So uh, so each time it has like an infections, it will the viral genome will somehow integrate it into the bacterial genomes, and it's like a mechanism to like to uh, remind about the infections, and it acquire the spaces from different infections.
Okay. Yeah. Right. Và Vân Anh Vân Gia Kiệt. So Thu Giang.
Yes.
So so how can we know um so for example for for bacteria, so they are different, they have many different spaces, so how can it Thu Giang?
Yes.
Yeah.
I can
So
hear you.
oh sorry. So for example for bacteria they have different spaces, right? So how they accommodate many different spaces, and what does mean for the bacteria?
Oh, sorry. Can you repeat?
Yeah. So I go here. So here, for example, we have too many different spatial layers. So that is an example. That's an example. But in nature, so the matter can have 100,000 spacer, different spacer. So how can the matter accumulate all the spacer? You got it? Okay. Uh, do you have any idea? No.
Not yet.
Okay. Maybe maybe Ngọc Hà. Can you help her?
Sorry, can you ask another one?
Oh, okay. So Đức Huy,
Yes.
did you follow the answer? Did you follow the question?
I have no idea. Sorry.
Okay. So here so you can see here we have different spacer in the virus genome. I mean no, in the bacterial genome. So how the bacteria acquire and accumulate different spacer. I mean many different spacer layers. How can it be, and what is the mean for the acquired system? Do you have any idea? Any idea or any general? You can say, yeah, it's fine. You can say what you think about, what you understand about. Say yeah, that's right. And so we will have some idea, so that yeah, we can explain if you don't understand. If you yeah say incorrectly, you answer incorrectly. So do we? No.
Does they use cast 1 and cast 2 as the helper for the spacer acquisition process?
How can you help? How?
Can I answer this one?
Yes, yes, yes.
Yes. So, like before, I didn't understand, but now I think I kind of understand. So I just want to check if I understand correctly.
Right.
So each spacer, as I understand it, comes from a different virus that has infected this particular bacteria before. And so after the infection and after the bacteria has integrated that viral DNA into its own bacterial DNA, then the viral DNA becomes that spacer. And the next time when a similar virus infect this bacteria, then the bacteria can recognize the virus based on the spacer that it already has on its genome, and therefore will break down the viral DNA instead of integrating it into the bacterial DNA again.
Anything else? No.
That's what I have understood so far.
Yeah. Maybe I want to know one more. Maybe Mingwes.
So I think in the in bacteria it has a cluster of spacer and repeats, and it's in when the virus infect two planes, the Cas complex translates to Cas 1 and Cas 2, and Cas 1 and Cas 2 cleavage the viral DNA. This fragment inserts to the new spacer units, and when the virus infects the bacterium against the charged RNA, translates to protein charge, and the Cas complex can translate into different Cas. It's not the Cas 1 and Cas 2, and then this is combined with the RNA polymer to the complex is a Chris RNA, Cas RNA, and Cas complex, and the cRNA is mean the spacer of that is a fragment of DNA virus. C inserts the cluster, and then this complex can recognize the DNA virus based on the guide RNA, and then it's cleavage the DNA virus into the small fragment.
Nutri.
Yes, I'm here.
Yeah. So why we have different spacer?
I think we have different spray in order to have different aims for RNA, for the RNA.
Anything else? No.
And I think it kind of it have different role. have different role in order to have the groups the groups K9 to the protein groups K9 to bind to the RNA the RNA and we had the enzyme when it completes and the enzyme will to complete the cut of it. I think so that, but I don't know. I don't know clearly about this. Yes.
Okay. Yeah. So I will ask someone, and after that we will talk a little bit more about it one. Okay.
Yes.
To be sure that everyone understand, everyone know about it. Hà Nhung, the same question. Mm. Anything else? No. Mm. Okay. Yeah. Yeah. I think someone understand very well. Like Phương Anh, yeah, Phương Anh understand very well about it. Anh Quân Vân Giang, but I think someone still, yeah, uh, yeah, still not very clearly because that's my problem. So I want to figure out and to know if everyone, yeah, got it. Ánh Xuân,
Yes.
mhm, the same question.
Sorry, I don't.
Yeah. My question was, why in bacteria we have many different spaces and where they come from, and so what does it mean for bacteria? I mean, how's the spacer helpful for the bacteria?
Sorry, I don't have the answer.
Okay. Is okay. You still have no idea, right? Okay. Minh Nguyệt.
Sorry, can you repeat the questions?
Okay. Sorry. So why we have many different spacer in bacteria, and where does they come from?
I think the
Where do they?
difference. I think the different spacer in bacteria like the either immunize memorized in the human's body. It means the bacteria can infect by a virus, and after each virus infects the DNA, the bacteria cut the DNA of virus, and then this can insert this DNA into the cluster, and after the insert, it can be it inserts into the space between the repeats.
Mhm. So where do it come from? Where do they come from? I
The
mean,
spacer,
right, the spacer.
the spacer come from the viral DNA.
Mm. So why we have different spacer?
Because the bacteria can infect by different virus.
Mm. Mm. Okay. Um, thank you. Big Ngọc.
I'm here.
Yeah. So, do you have any addition for spacer, and do you have any idea about the mean of spacer for the virus and for the bacteria?
Uh, the Nhung has said that the spacers is from the DNA, which is cleaved from Cas9 Cas enzyme to the viral DNA, and I have the same answer with her about that. The spacers come from first is come from the virus, of course, and the bacteria have many spacers because it has to remember the infection from different the virus, because I think that the virus have each virus have a different infection for the bacteria, so the bacteria have to remember the DNA of into the spacer. So when the old infection occur, so they can recognize easily. So I think that is my understanding.
Right. Right. Okay. I think most of you understand very well, and someone still did not get it. So uh, I think Vân Anh, no, yes, Vân Anh. I think Vân Anh understand very well about this. So can you repeat once again, like explain for everyone once again?
Yes, of course.
Yeah.
Okay, so the first question, why bacteria have so many spacer? So spacer come from viral DNA, which means when the virus infect its DNA into the bacteria can clip the viral DNA and integrate that into its own genome. So the viral DNA is spacer. Why bacteria have so many spacer is because there are so many types of bacteriophage. So that's why we have so many spacer, and each time a virus infect the bacteria, there is in its genome will stay in the bacteria, and the bacteria collect viral DNA through time. So that's why in the bacteria we have so many spacer, and how the spacer mean to the bacteria after they integrate the viral DNA to be a spacer. The spacer can be transcribed into a guide RNA for the CRISPR complex, and when the same virus infect the bacteria, this guide RNA can recognize the viral DNA, and it lead to it guide the complex, the CRISPR complex, to clip, to destroy, to break down the viral DNA, so that the bacteria can survive from the infection.
Right. Right. So yeah, thank you so much, Vân Anh. So I think Vân Anh Quân, and yeah, Phương Anh, yeah, understand very well about this. So I think we move to another part about the structure of the CRISPR RNA.
Sorry. I'm sorry.
Yes, go ahead.
Can I ask?
Yes.
I have some questions.
Yes.
So where's the protospacer? Yeah,
Photospace.
and we know that the spacer is from the foreign virus from each time of infection. I want to ask, how do the bacteria, how do they know which one they will keep, or they cleave all the spacer?
Oh, right.
Yeah.
It's very interesting question. So because I mean, um, this this occur randomly. So it means that it's naturally. It's not like selection. So whenever I go back here, so whenever we have virus DNA, yes, when the virus inject their genome, normally we have RNA, and RNA convert into the DNA. Yeah. So we have a CAT1 and CAT2. So it's the enzyme. It's a nuclease enzyme, and it breaks down the DNA, and it breaks down to many different fragments. And that fragment insert into the spacer between the uh, repeat region. Insert into this region between them. It's randomly. It's not like specific insertion. So, and after that, we have different spacer. So therefore, we have different kind of RNA. So, so for for acquired season, for future infection. Sorry. So when the same virus infect into the bacteria. So now the bacteria have spacer that can recognize different side of a genome. A virus and a viral genome. So it can break down the specific virus in different side, different side of the genome. Yeah. Is that clear for you?
Yes, it's clear.
Yeah.
One more question.
Yes.
Yeah.
Okay.
I want to ask whether the trace RNA is species specific, or it is the same in all kind of bacteria,
Right.
and do we have the same mechanism like the CRISPR Cas in our human body? Do we have this?
So the first question about do we have common trace RNA or we have specific trace RNA? The answer is we have universal. So mostly we are universal trace RNA. The trace RNA is quite constant. It's not variable between. It's not. So of course it's different between bacteria to bacteria, but in the same species it's common and it's constant. Yeah. And for second question, do we have the same mechanism with same system in human body? So the answer is no, we don't have it. Yeah.
Okay, thank you.
Yeah. Anything else? Any question from any student?
Can
No?
you say it again about the universal trace RNA?
Yes. So the universal means that the RNA is quite common for. It's common for all RNA because. So actually, I should mention about the RNA, how the structure of RNA, but so the lecture is go by like this. So it's something like I go into the I go to and I go back. So yeah, actually we don't have it. So here we have RNA. So this RNA is common for almost RNA. So the RNA it contains trace RNA, it contains CRISPR RNA. It had two rhythm, and also it contains the pomo spacer. Pomo spacer is come from, I know it's normally it's from 2 to 6 different nucleotides, but normally we when we design for example, we design RNA, we design only three promo spacer. We call PAM. Yeah, we call PAM. So I think I had to go to next slide to for either for you guys to understand about spacer. But I can explain a little bit here, and I go to very detailed in another slide. Okay, so this tracer come from this, and it it synthesize it transcribes independently with a spacer. The spacer that only transcript to the CRISPR RNA, the CRISPR RNA, and then combine with the tracer RNA, and it form the guide RNA. Yeah. So actually, for guide RNA, we have two main. I would say two main component. We have tracer RNA. So we have tracer RNA here, and we have CRISPR RNA transcript from different spacer. Yeah. So basically, and of course we have space, and we call PAM, but PAM is very simple. So Vân Anh.
Yes, I understand. Thank you.
Okay. I was mentioned a little bit. So spacer is have many different hairpin. It has hairpin structure to form special structure of tracer. So it's so actually so normally it's when before I mean before forming this structure is that the stress is the stress RNA, but because it have some complementary nucleotide, so it can bind to each other. It can so it has and it forms the cell dimer, cell hairpin. So it has the hairpin domain like this. Yeah. So it have scaffold like this. So that helps to the CRISPR. I mean the Cas9 protein can bind to this tracer RNA, and it can recognize this one, and after that it can activate it. Yeah. If the tracer RNA cannot form this, for example, this form, so the tracer RNA has no function. So therefore, the guide RNA has no function. So results, the Cas9 protein cannot bind to the guide RNA, and they cannot cut anything. Yeah. So so here we have tracer. So tracer is encoded by a specific gene that encodes for the tracer. I mean universal tracer only, and we have different post space. It's like spacer. So spacer that we mentioned before, it come from different infection, come from different virus, and so we have different CRISPR RNA, and after the cell transcript, I mean it converts CRISPR RNA and tracer RNA. So that two different RNA bind together, and it forms the guide RNA. Yeah. So here, so the CRISPR RNA and tracer RNA, yeah, and it's they only bind together by a specific reason. It's content reason. So this content reason is so it's a repeat reason. So you can see it transcript from the transcript reason. This and this, and it's common for CRISPR all CRISPR RNA. So that's why the tracer RNA can bind to that reason. Right. So and we have this complex, we have tracer RNA, and we have CRISPR RNA. So after that forming, we have guide RNA. So right now we have guanine, and before this step, we don't we don't call we don't call guanine here. We don't call it. Yeah, because guanine is formed by two different components. Sorry, in nature, and actually in so because we learn from nature, we learn from bacteria, and so now we have technique to do that, to form the guanine. I mean, we have to form guanine, and so like few years ago, yeah, more than 10 years ago, we normally people use single guanine. So it means that so this one will be encoded by a gene that link between guanine and tracer RNA, and it's linked together. It combine both guanine and tracer RNA. It's not two part guanine. Yeah, I will mention in the next slide. Yeah. So here we already cut about how bacterial DNA can be digested by the virus. Yeah. Can someone mute? Someone mute green. Okay. So here we have so this is a structure of CRISPR, of Cas9 protein. So we have different domain, but I would like to say two important domain. So we have a SNS domain, and we have IUVC, and we can say RUFC domain. So this two domain can bind to. Um, it contain nuclease activity core. So this one can bind to the DNA of the uh, I mean a viral, and it can digest the viral DNA by the two domain. So if we have some mutation in the two domains, so normally no, so we have no function. The Cas9 lost the function, and even it can bind to the DNA, viral DNA, but it cannot cut or cannot digest the viral DNA. So the key important is the two domain SNS and UVC. And we also have the PAM interaction. So I mentioned about the PAM before. So PAM basically we have from two to six nucleotide, but normally we just need three nucleotide. That enough uh, for the canine protein can recognize the specific reason to create the DNA, the specific reason that we need and to create them. So we had different domain, but you just remember three very important one. Yeah, we have this one and this one that contain nuclease activity. It has endonuclease activity. This one, and we have PAM domain that can recognize the PAM sequence in the guanine, and so as I can prevent the DNA. So here's how I would like to show you how people RNA work and how it can digest the DNA. So the CRISPR complex we have we have Cas9 protein. So this Cas9 protein, it look like ribosome, right? So it have different domain. So as I mentioned, we have S and S when have a, so we see, so that two reason can recognize, I mean, it have a endonuclease activity, and it can digest the deny a specific reason. And we have spam sequence. Spam sequence, as I mentioned before, we have 2 to 6. So here, so we have 3 nucleotide. So it's very important. Is that only 3 nucleotide? So we normally we have NGZ. So N it means anything. So it can be T, can be A, can be C, or yeah, whatsoever. We have 4 different, right? N can be anything between 4. So among 4, uh, and so this reason helps the keeper. I mean the cat9 can bind to and can recognize the specific reason and to cut a specific reason. And so we have how to say, we have guide. It's not guide. It's, I would say it's the guide sequence. Yeah. So actually, it belongs to CRISPR RNA. So it have a guide sequence that can recognize specific sequence. So normally, the guide sequence is reading from 16 to 20 nucleotide. But when we design CRISPR-Cas, we design something like 19 or 20 nucleotide. And so in CRISPR-RNA, we have a content region. So this content region can bind specifically to the tracer RNA. So the tracer RNA. So as I mentioned before, tracer RNA is like universal RNA. So it can bind to any kind of CRISPR RNA. And so in CRISPR RNA and in tracer RNA, we have scaffold. This scaffold, it have multiple hairpin point, so they can bind themselves and to form the scaffold to form the structure. So that help the RNA can recognize the RNA, and it can activate by this specific uh, domain. Yeah. Any question for this?
Can you explain again the function of palm protein, a palm domain?
Yeah. So so actually, palm domain is contain only three nucleotides, and it's a sequence that required for Cas. I mean, for Cas9, Cas protein can recognize and to create the sorry to create DNA sequence a specific reason. Yeah. So normally, when the Cas9 complex bind to the spam sequence for this reason, so it lets the Cas9 know how to where it can create the RNA. So normally, create the DNA. So normally, the creates that occur that before the PAM sequence. Yeah, the PAM sequence that it help to the canine protein determine where they can cut the DNA. Yeah, and the guianated one, this sequence, they help to the complex. The complex can bind to specific sequence. Yeah.
Okay.
You got it? Yeah, you got it?
Yes, I got it.
Okay. Anyone? Any question? No?
So, what if the target DNA don't have the PAM motif? I'm not sure that the PAM would bind to the target DNA, right? No.
The PAM sequence is, it the palm is in the genome. Yeah, it in the genome. You got it?
So the target DNA is viral DNA, right?
You mean this one?
No, the yellow, the yellow.
This one, right?
Yes, but the yellow sequence, the yellow,
This
the
one?
palm is orange. Yeah.
This one, right?
Yes, yes, yes. Is viral DNA?
Uh, you mean this? You mean this?
Yes.
Yes. So actually, the palm stay on the, on the genome. Yeah, on the viral genome. You got it?
So the viral genome had palm.
Yes. So actually, palm very simple. Is that true? Some reason that contain palm. For example, when you design CRISPR, you design RNA, CRISPR RNA. No, not CRISPR RNA. CRISPR RNA. So you design this. You choose this sequence, and you need to determine where in the genome, in the viral genome, have palm sequence. So it that pick up some sequence before palm. So the palm stay on the DNA genome. Yeah, you got it.
What if the palm is so far away from the guy DNA?
Right. So for example, if you design the RNA that's so far from the palm, for example here, so it cannot recognize. So it fail. The whole complex, this whole complex fail to activate, and it cannot cut the DNA. It can divert the DNA. It cannot activate. Yeah, because for the all activation, it needs palm. Yeah, and it needs the RNA. So actually, I would say guide sequence, and it needs the tracer RNA to activate the Cat9. Yes, and without palm and without uh, guide sequence, so it cannot cut. Yeah,
Okay,
you
I
got it.
understand. Yes.
Yes, yes. So actually, palm is not on the genome. Yeah, but when you pick up the guide sequence, is that true that the palm need to dry up to the guide sequence? Yeah, that near to this one. Mm. Someone palm military prospacer. Prospect. Another 2 or 3 base can locate within or located that dream. Oh, yeah. So yeah, it's right. But I mentioned that when you design, I mean, when you design a CRISPR-Cas, a RNA, is that determine that you have that PAM? Yeah, it can be, and I mentioned it can be from 2 to 6. Yeah, 2 to 6 nucleotide. But for optimize, yeah, right.
Okay, so I want to write that because she asked if palm can be far away, but because the definition, it first of all it cannot be far away, and second of all, as you mentioned before in the Cas9 protein, palm cannot bind to the guide RNA. It binds to the palm interaction domain of the Cas9 protein.
Right. Right,
So
right.
this Cas9 protein found the palm and interacted with the palm interaction domain, and so
Right.
then put the guide RNA in, and then start the cleavage by HNH and the other protein.
Right. But it cannot be too far away, for example, like somewhere here. So it cannot. Yeah, it cannot work like this. So you see what my point? Quân Anh?
No, I don't have any issue. I just want to answer the question for her. That's all.
Quân Anh. Okay. So any question? Vân Anh?
No.
Okay, thank you. So so now we mentioned about Taiwan. We mentioned about different cat 9, and we already mentioned about how to say cat 1 and cat 2 before. So now we go to to have a general idea about different type of keeper cats. So actually we have many different kind of keeper cats, but we just focus on some very common and some that helpful for for future application. Yeah. So so we have like cat 1 and cat 2 is class 1 and class 2. So it's not very common in Zin. In editing. So I think I will not talk more detail about it. So I mentioned about cat 1 and cat 2. So if you remember about the first slide when we discussed about how how bacteria can prevent viral infection. So actually we have cat 1 and cat 2. So cat 1 and cat 2. So cat 1 and cat 2 is that universal nuclease. So it can digest like any kind of DNA. It's not specific at all. It's that DNA and it digests all types DNA, and also can sometimes it can digest RNA also. And we have cat 3 because cat 3 important in DNA synthesis. Do anyone know the function of DNA helicase in the bachelor study? In undergrad. Quân Anh? Do you remember the function of DNA helicase? Quân Anh?
Vân Anh? Helicase is the enzyme that can bind and remodel the nucleic acids to unwinding unpack the organism gene.
So what is the function in the cell? When the cell needs helicase.
DNA replication.
Yeah. And how about endonuclease? The function of endonuclease and how it works. Yeah. So do you remember exo and endonuclease? What is the difference between exo and endo?
So as I understand it, the nuclease, it can break down the bond between nucleotides. And about the endo and the exo, I'm not sure. Yeah.
Is that yeah? Go ahead and so.
The endonuclease is the enzyme that clips the polynuclear chain in between, while exonuclease refer to enzyme that clip polynucleotide from the end of the chain.
How does it work? How does it work? But different. I mean, very different between endo and exo. So the way they digest the DNA. How?
Endo cut in between and create oligonucleotide, while exonuclease cut from the chain end, so cut one by one. Right.
Okay. So anyone, any Đức Huy? Me to want to say something?
I remember EXO is the cut tie in the I mean in to no I mean in the point start, or however in the end endo they cut in the how I can say. I mean the difference is the position to enzyme cut on the DNA.
Okay, I need another question, another answer, another idea. Nam Khánh hay Nam Khánh?
Nam Khánh. I don't remember the function of exonuclease.
Linh, Thị Linh, Nguyễn Thị Linh.
I think it's the difference between them is about the capillary position, but I don't remember clearly about this.
Okay. Tuyết Mai.
Yes, I think that the difference between the endonuclease and exonuclease is about the site of the cut site, the cutting site. Like with the endonuclease, we doesn't need it doesn't need the three or five, like 5 phosphate or 3 hydroxyl group free to activities, but is needed by the exonuclease. That's why the endonuclease can cut the inside of the DNA change. The inside of the why the exonuclease cut in the end, into ends of the DNA. That's I remember.
Another question for everyone. Do you think that exonuclease can cut the whole sequence, or it cuts like few nucleotides? It can digest the whole sequence, or it can digest just few nucleotides. Any ideas? Salmai?
Yes.
Do you think it can cut the whole sequence, or is that
The exonuclease.
Yes.
I think maybe they can cut the whole sequence because they cut at the three prime, and it's like the two end of the DNA molecule. So maybe they can cut the whole. it maybe it's not whole, but it's more longer than the other, like the endo or the other endo.
Yeah. Okay. So yes, I think some of you answer correctly. The difference between endo and nucleus, I think who is that Samai before? Yeah, I think she answered very correct about the why and how how the isonucleotide can cut and cut the cut the DNA a specific sequence. And normally endo and iso are quite different, like the way they cut the DNA. Endo can cut it cut in the middle of the DNA strand. It can cut anywhere when it recognize a specific reason. And is that that's only point. It cannot cut the whole thing. But for the EXO we have two different EXO. We have 5' and 3' isonuclease. So all 5' and nucleate and 3' isonuclease it can cut the whole sequence. So normally we have some polymerase that contain isonuclease. So it can cut by for example 5' isonuclease or 3' isonuclease, and after digest the DNA, so they can they can repair. Normally for some pone, for example, for pone 3, for example, they have two different activities. They have iso activity, no, isonuclease activity, and they also have polymerase activity. So before they digest the DNA, and after that they repair by adding the DNA sequence. So that is the mechanism when the cell repair, min-max repair, or they can repair the mutation. Yeah. So so that all cut 1 and cut 3. So you guys know all about it, but that different name. But the mechanism and the kind of enzyme you all know in the undergrad. So and cut 4, cut 5, cut 6, cut 7 is not very common in study. Yeah, not very common. And so now we mostly focus on cut 9, cut 10. Cut 10 is also not very common. Yeah. But now we have cut cut 9 mutation, and also we have cut 12A. Cut 12A also important. Yeah. But in this slide we don't have that. We did not mention about that one. So so basically the most study from now like last decades since 2012. So when Emmanuel and Jennifer propose the mechanism, propose the CRISPR-Cas technology for genome editing. So like we have like few thousand study focused on CRISPR-Cas9. Yeah, and we have like few thousand publications from 2012 until now, and most of them focus on CRISPR-Cas9, which were isolated from Streptococcus pyogenes. So for the summary, so as mentioned about for CRISPR-Cas complex, we have firstly we have RNA, we have trace RNA, we have farm sequence, and we have so that's farm sequence belong to belong to the xenomania, and so we have canine protein. So why we say three component for CRISPR complex. So basically we just have a canine protein, and we have a guyanine. Guyanine contain, yeah, I mentioned before, it contain uh, guy it, guy sequence. It contain tracer RNA, and so it have PAM. So PAM sequence stay on the genome RNA. So here, so for the CRISPR-Cas complex, we have Cas9. So that's the first component. So Cas9, and we have second component is the guanine. The whole thing that is a guanine. So guanine contain guide sequence, and it contain tracer RNA. So that is the second component. And the third component is PAM sequence. So PAM sequence stay on the genome of the virus. Yeah. Yes. I mean viral DNA. Yeah. So that is three components. And so I think you all know about defense mechanism that help the bacteria to prevent viral infection. And yeah, we know some, I mean, some CRISPR-Cas system, but I want to stress about, I want to highlight about some CRISPR-Cas system that we used to in research now. So again, we have Cas9, and we have Cas9 mutation for different purpose, and we have Cas12a that very common now, and we also have a Cas9 that that use that can use for multiple DNA. So we call nickase. So nickase also a Cas9, but yeah, it have a different mutation. So it have to the Cas9 that recognize, and so to cut the DNA not like double strand, but it's that cut only single strand DNA. So I think I will talk more about that in next few slides. So for the CRISPR-Cas technology, so as I mentioned, is it was study in CRISPR-Cas9 were booming since 2012. It's firstly proposed by Emmanuel and Jennifer, and so both of them were win Nobel Prize Nobel Prize in 2020. So from this discovery, it lead to multiple application right now. So we have different application in bacteria, in drug development, so in also plant biotech, and so even in human. But human, so we still have a lot of controversy about CRISPR-Cas9. So so again it's so the one that is not, so I think we don't need to repeat about it one, because we already cut about this, spend a lot of time about this. And so now I would like to talk a bit about the engineering. How can we use CRISPR-Cas in the real experiment, real research? So so normally we have two ways to make CRISPR-RNA, to make RNA. Sorry, didn't see anything. Hi.
It quite small. Can you zoom the picture?
Can you see it? Can you see it, everyone?
Yes.
Okay. So so actually that is the way how we decide how can we apply CRISPR-Cas9 in the real um, uh, I mean research and the real experiment. So normally to have RNA we need CRISPR. I mean we need CRISPR RNA, right? And we need tracer RNA, and we to um, to let the CRISPR-Cas system work. We need also Cas9. So in some, I mean in for example, when we want to target in uh, animal cell or in plant cell. So in plant, I mean in general, in eukaryote cell, we don't have CRISPR-Cas system. So even in some kind, I mean in some bacteria, we also don't have CRISPR-Cas. It's not like all bacteria have a CRISPR-Cas system. I mean CRISPR-Cas9 system specifically. So therefore, we need to engineer. We need to contract. Yeah. The I mean normally we use some plasmids that we insert the gene sequence of interest. Normally we insert the gene sequence that can encode for CRISPR protein, and we need to encode for RNA. Yeah. So um, for some, I mean for some application or some study, we can use like multiple CRISPR-Cas. I mean multiple RNA in the same plasmid in same structure. So uh, so therefore in there's only one. I mean one plasmid construct. When we insert into the cell, it can generate different kind of RNA. For example, here we have DFP1, DFP2, 3, 4, and 5. So actually, DFP that are the gene reporter that we can know. We insert into the cell and we know the expression of different gene. So DFP is not special. It's not special in this case. So is that the example for explain how can we, yeah, we can design. I mean, we can design the RNA, different RNA, and to target different sequence in to target the cell. Uh, is that clearly for you? Fernand?
Yes, I understand.
Yeah. Okay. So so now we move to different kind of RNA. So as mentioned before, we have a single RNA and we have a two complex RNA. So two complex RNA, that is a natural way that I mentioned before. Yeah, I already mentioned before about two RNA. But so here we have single guanine. Single guanine is mean that from the guanine have a unit sequence. That unit sequence that contain both tracer RNA, I mean trans activating creeper RNA and palm no not palm and creeper guanine. So so I would like to come back here. So here we have two form of here we have two form guanine, or we can say double plug guanine. So we have separate creeper RNA and we have separate tracer RNA, and they on bind together by tracer RNA bind to creeper RNA by content content sequence. Yeah, that we call the universal sequence that can bind to all creeper RNA. But so in this form, we have we have single guanine. Single guanine normally is not it not exist in nature. It's not common in nature. So single guanine that we use for engineering, and we combine two sequence. Yeah, we uh, we design a gene that express the fusion creeper RNA guanine. Yeah. So here, so for the single guanine also similar. It also have the scaffold that's formed by different hairpin structure. So even it have different, it have a different nucleotide can by complementary to each other. And so this one have to activate the canine RNA. And so we have the palm sequence somewhere here. I mean, not palm sequence, but we have palm structure on the canine that recognize palm sequence on the genome RNA. So here, so they go all similar. They go similar as two form. Two form guanine. So it's quite similar. So when the guanine recognize the sequence of interest, the specific sequence, so it leads to digest the genomic DNA of the viral. Or so in this case, it's not the viral, but maybe the human or bacteria, plant cell or animal cell. Okay, let's see. Okay, uh, we come back about two mechanism of double strand repair. So as we mentioned before, I mentioned before. So we have two ways. Like we the cell can after after digest. After DNA digest by the enzyme. By triple Cas complex. So it leads to have two different things. We have like sorry. We have double strand. We have double blunt DNA, or we can have overhang DNA. So when we have overhang DNA, it leads to the cell goes to homologous endoid repair. So in this way, it's that insert or it then leads the sequence. Yeah. In this case, yeah. Firstly, it need to, the cell need to repair the overhang and to make the blunt DNA, and then it can insert a sequence. A DNA sequence into. Oh, sorry. Can we take like 5 minutes for break?
Yes.
Okay. So for for this pathway with that we have indel. Normally we have indel. Indel it means that we have insertion or we have deletion. So it means that the cell can insert. I mean, oh sorry, it can insert some DNA sequence or some nucleotide in that region. I mean, after cut it and it can insert some sequence or delete some sequence. So we have this pathway. And for homologous direct repair. So when we have a blunt sequence, and so in the I mean in the I mean in the cell we have DNA from we have donor DNA. So donor DNA can come from in nature. It can come from different. I mean, for example, from the DNA breakdown, or it can come from I mean different digestion like viral DNA digestion. But in technology, I mean in research or I mean for human purposes. So we have DNA, we have donor DNA that uh, we put into the I mean into the mixture. So normally for in research, so we can do in vitro experiment. So we can you can introduce the donor DNA into the cell. So so after breakdown by this pathway, so the donor DNA yeah can work by replacing I mean replace the whole sequence, the same sequence because we have some permutation in this way, and we want to edit it. We want to repeat the right sequence. So we want to correct the sequence. So and so we use that, and we that's why we use this pathway. And so we have two things to do. It can repeat the whole thing, or it can just correct some. I mean, correct some nucleotide. Yeah. Any question for this? Some I understand.
Yes, I understand. I want to ask why in if we give the donor correct DNA, how can we make sure that the our donor DNA can insert in the genome, or we just
Right.
it will occur randomly and we will check it later.
Right. So actually, the efficiency for homologous direct repair is very low, super low compared to this pathway. So for example, in this pathway we just have a 1 to 2% efficiency, only 2% efficiency. But in this case, yeah, for example, we sometimes we can got more than 90% knockout. Yeah, 90%. I mean the efficiency in this case very high. But in both case we can check um for example so here we can check by sequencing because after after knockout we just do PCR, simple PCR, and we amplify the sequence of interest by and after that we can do like single sequencing. Yeah, it's super simple by this way. And for by this way, normally we can do like we can do. We don't need to know the specific sequence. We don't need to know the sequence But we just want to focus on the the function. I mean, the actual function of the in of interest. For example, I want to knock out PD-1 gene, for example, and so it's very. I want to know if PD-1 would knock out. I just do immunochemistry. So I just stain the cell with a specific protein and antibody that binds to PD-1, and then I can do flow, or can I can look the cell under microscope, or I can do flow cytometry, and so I can know the knockout cell or the white tie cell. Yeah, it's super simple to track this one, but in this one sometimes with that correct I mean with that train only one nucleotide, and it's very difficult to to detect it. Yeah, because sometimes you cannot detect you cannot detect editing by flow cytometry or immunochemistry. Yeah, because with only one nucleotide, because sometimes it cannot change the I mean frame shape. It cannot change the open reading frame. Yeah, the mechanism is, so when you insert or you delete anything from into the gene of interest, the sequence of interest. So all the sequence after this point will be trained. So all coding, coding will train. So that's why all peptide, all protein will train because it train the codon. Yeah, train everything. Even if you insert just only one new, or you delete only one new, all the frame shape will be trained. So we call open reading frame. Yeah, it trains the whole thing. So the protein have no more function. Yeah, it also convert. It also synthesize. The cells can still synthesize the peptide, but the peptide will be different because the form will be different when the protein post translation it goes to the Golgi, right? The Golgi complex, so it formed differently. So the protein no longer the right structure, and it had no function. So so actually we have we have different kind of in not CRISPR system, but we have different kind of technology uh, to use for using for gene editing. So the most common in the past is gene finger. So this one normally we use for animal. Yeah, for knocking or knockout gene in animal. So talent a um, talent is different. It's not recognize the sequence of interest by the RNA, but it recognize the DNA sequence by different amino acids. Yeah, so we have specific amino acids that are recognized for specific nucleotide. So that are different between talent um, and CRISPR-Cas. But for talent is it's quite easy to design and it works very specific. So talent normally used for knockout or knocking in animals. Yeah. And CRISPR-Cas9, we already mentioned about CRISPR-Cas9. Um, yeah. So here, this is the structure of CRISPR-Cas. Mm, yes. So it says that we require only two nucleotide. So basically, we can design from like 16. We can design 16 to more than 20 nucleotide for the guide sequence. Yeah, but normally 20 nucleotide is optimal. Yes, that is optimal length for guide RNA. Yeah, but for CRISPR-Cas, we also have some limitation. the most difficulty thing is off target. So it doesn't mean that you have specific guide sequence that by that recognize a sequence of interest, but sometimes it can by randomly to different side of genome, and we don't know when it by uh, incorrectly, I mean non-specifically to the sequence, so it can cut different in different side, so that is a limitation. But so now we have the new technology to to uh, to control that. For example, we can select different for cell line. We can different cell line based on next generation sequencing. After knockout, we can verify the cell by whole genome sequencing, so we can know which sequence that have a off target knockout. But um, but actually, we when we design, we have different tools to see. I mean, to control the off target and on target sequence, so we can avoid that problem. Yeah, basically we can avoid. And further, when we work with cell line, we also need to have two different guideline, different independent guideline. So it means we have two independent clone, so we can compare, we can have control. We have internal control, and we have white type control. And when we want to study the function of one specific gene, we can have different control. We have white type, we have two different clone from two different guideline knockout. Uh, from the same gene, so we can to control that problem. Yeah, normally. So he also mentioned about this. So for so I mentioned about, I mean, sometime before, like so CRISPR-Cas9 that I mean the application of CRISPR-Cas9 that begin from 2020. I mean it was booming from 2020. I know 2012. So from 2002, that's only we have some study about in eukaryote cell to prokaryote cell to mention about they mention about the decline. They talk about the repeat sequence, and they don't know what is the mean of this sequence for the bacterial cell. And after that, in 2012, as I mentioned, when Jennifer and Emmanuel focused on CRISPR-Cas system, and they propose the technique for gene editing. And after that, after their publication, so we have, I mean, we have thousands study focusing on this. So now, for example, in 2015, so it's very old data. So we have more than 1,000 publications related to CRISPR-Cas9 compared to like few publications in early 2000. And so the investment for CRISPR-Cas system also increased gradually, not gradually, but over, I mean, the last decades. And so actually in 2014, so there are about 160 million investment focused on CRISPR-Cas9 and iPS cell. So it's iPS for induced pluripotent stem cell. So now I would like to introduce very basic about the application of CRISPR-Cas9 in research and development. So we have drug discovery. So actually, in our department, we have Dr. Tuấn Anh. Actually, he worked in CRISPR and he so he actually used CRISPR system to screen different drug targets. I mean, yeah, because uh, so we can quickly I mean screen some drug target or candidate for future research using CRISPR-Cas system, and we use CRISPR-Cas for animal. I mean, for knockout or and for knocking something as I mentioned before. Yeah, and we also use for plant biotech and also for gene editing. So actually, gene editing we have some study in human. So anyone have any idea or any knowledge or any updates about human gene editing recently? Last few years, in the last few years. Quân Lanh,
You ask me?
yes, yes.
Can you repeat the
Do,
question, please?
do you have any update about human gene editing using CRISPR-Cas system?
Um, I think there are one in China, but they are banned at the moment.
Right. So how it was? Do we have
Well,
any?
if I know, if I remember things, that there was this story Dr. Ming has already said to us. One is about scientists in China that edit that want to make the perfect children. So so he decided to do genome editing on the children so that they won't receive cannot be transmit HIV.
Right.
But it is not ethical. It is unethical. So he had been put in prison. Even though, from my perspective, I think that his work can be good for the future, but but at this time, it's just not ethical and it illegal. So yeah, it's not gonna work right now.
Yeah. So yeah, actually, the reason that why they want to edit it, the scene, because they see that in about they see that in EU, I mean, or in northern countries. So so there were about 1% of patients who have mutation in CCR5 protein. So the mutation in CCR5 protein lead to resistant to HIV. So therefore, they want to knock out that gene and to see if so the patient can I mean the I would say the patient can resistant to the HIV HIV virus. So actually, in study from one group from China, so they yes they start with two different embryo. So yeah, and they do keep a cat night. I mean, keep a cat knockout in that two embryo, and so from one embryo yes they got they get mutation in I mean they got India mutation in one strand I mean one locus and another locus they don't have any mutation, so it means that they have a heterozygous embryo I would say baby. So that's mutation. So probably cannot resistant to HIV. And another embryo, so they have mutation. They have indel and they have yes, they have a indel and they have a insertion of some nucleotide. They have big indel and they have some small insertion. And so that's that embryo will develop into a baby, and so technically that baby can survive from HIV, so it can resistant to HIV. But the problem is uh, different study published in Nature indicates that when patients who have a mutation in CCR5 gene normally have a short-term survival. Shorter life than the naive. The white time. Uh, yes. So that's why we still have a many questions about this. And yeah, and now study in human. So in human, actually, in like human body is still very limited. Yeah. So actually, people hope to can use KIPA-Cat9 to treat some kind of cancer, for example, blood cancer. Yeah, but they still yeah unsuccessful because we have some problem with off targets, and we also have problem with efficiency. Low uh, gene editing efficiency, and we still have problem with how to maintain the muted cell. Yeah, maintain the edited cell. Yeah. So I think we need more time to discuss about that because it's very long story, and it's related to different technique, different technology. Yeah, because when we want to mention about gene editing in treatment, we mention about cancer, and we also mention about the the cell therapy. So this therapy will be another story. Yeah. So actually, technically, we can focus on some field, but I think in future is very uh, promoting a drug development. I think it's very good thing. Animal study for gene knockout, knocking, and to have some model. Yeah, animal model. I think it's very promotive and no promising, and it can be used for plant biotech. Yeah, plant biotech is very, yeah, I think it should be the good target. And for material, I don't think it will be, yeah, it will be helpful because I don't see any, yeah, benefits. Um, yeah. So now for the general material that we need for KitKat technology. So we have different cell lines. So actually, this cell line is that used for KitKat study. So it's very well. So we have hack 293 T cell. So actually, hack we have different kinds. Hack 293 T cell, 293 2 293 FT, and we also 293 and whatsoever. We have different kind of this one, but this one is come from human embryonic kidney cell, and it have some mutation, so it can keep the cell. Generate for a long time. Like it's quite similar like cancer cell, but it's not cancer cell. So we use this cell to make different thing for kit podcast. For example, we use it one for contract. To synthesize some plasmid or kit podcast structure. I mean, some RNA, and we can also use it one for lentivirus because we can use it one for add the hot cell for lentiviral production. So for example, if you want to use single RNA, you can use hex cell. So the hex cell can produce the virus, and after that we have virus that contain the sequence of interest. So actually it contains the sequence that can encode for single RNA. Yeah, and that single RNA can. So when the virus infect into the cell, and it can express the single RNA, and after that it can do knockout. So actually you can have you have different cell line, but I highly recommend to focus on hack 293 T cell, 293 cell, because it's amazing cell that used for different things, for lentivirus production, for Cas9, and also for produce adenovirus. So yeah, so in our lab we use hack 293 T and 293 FT cell. Yeah, for different purpose. And if you we use that for cell therapy and for CAR T cell. So CAR T cell, if you want to know more CAR T cell, maybe we can discuss about it later. So, but it's amazing. And for the model we have of mice, rats. So rat is not very common because rats is very big and it's more difficult to maintain. And we have fruit fly, but they are also not very common. So not very common. So for application in plant biotech and in no, I don't think I should mention a lot about it one because we still have very little study about all this. And so as I mentioned about the biomedicine, so we have iPS cell. So actually, so when you have lecture with Dr. Ming, you can ask her about iPS cell because her lab work a lot about iPS cell, and she a very good experience about iPS cell. Yeah. So that is the main. I mean, some key topics that we use Keepercast system. Yeah. In research and also in the real application. So for application, so we can sorry. Yeah. Okay. So we CRISPR-Cas9 to knock out or knock in culture cell line. For culture cell line, it's very easy compared to primary cell. So primary cell it comes from the human, or it come from animal. So it's it have many differences that uh, we that very difficult to work with keeper system because because they cannot maintain for a long time. Firstly, they cannot maintain for a long time. They cannot explain for multiple time, and it also has special memory structure compared to cell line, because cell line will adapt with the culture condition. The initial culture condition, and it can survive for forever. It can divide forever, and so it have for example some cell line it changes their structure, so it can, for example, some cell can bind to the culture disk, bind to the flask. Someone cannot bind to that one. So they have special memory, so that either to infect for example, infect plasmid or also infect the virus into the cell. So yeah, we can use tuberculosis for knockout or knocking in animal. So as I mentioned before, we use for some animal we generate for some uh, animal model. So for xenopic, so we still have some limitation. So I think I had to go quickly because we don't have we don't have enough time. So in general, we have different form of CRISPR-Cas. So for example, we have indel, like the way that CRISPR-Cas work. So indel that means that we have insert DNA sequence, or we have deletion. Yeah. So we call indel. So indel is the most common application for CRISPR-Cas system. Yeah. So for or we have replacement. So this one we as I mentioned before, we have homologous direct repair. So that is a second application. But this one, as I mentioned before, we have very low efficiency, low gene editing efficiency, and for large deletion. So this very important that I mentioned before. So normally when you want to delete a big sequence, I mean a large sequence, so it is different guiana in different position. If you have only small guiana here, you just delete small fragment, and so in some case you cannot. Yeah, in some case you want to, for example, you want to verify knockout by simple technique. For example, you just want to the uh, knockout efficiency. So and also you want to test the knockout cell. So you need to, but you don't have a like material. You don't have fancy, modern equipment like flow cytometry or immunohistochemistry, machine microscope or anything. So you cannot verify the knockout. So you need to do the big knockout, big the large dilation. So you can verify the knockout cell by simple way like by electrophoresis, just to do agarose electrophoresis. So it's very simple. For example, that is one example for verification and for application of this one. So this one is benefit for some library no for some labs that don't have a well equipped I mean equipment. And so this one I mentioned about CRISPR mutation. So when CRISPR-Cas, the Cas9 actually Cas9 have some mutation on the domain. So normally it have mutation in two domains that I mentioned before. So in two domains that have a nuclease activity. So it cannot knock out the gene. Even it can bind to the sequence of interest. So it have every it have guianate, it have the guide sequence, and it have everything. So it can recognize the sequence of interest, but it cannot delete the gene, it cannot digest the DNA. So we use that system for gene activation, and to use that CRISPR-Cas system as a enhancer to overexpress some protein of interest. So that is the application. And another thing is quite similar for this one, but we have like we have multiple guiana. So we have multiple guiana, and we have the gene reporter. So in this case, we have GFP, for example. So GFP is that protein that can emit the fluorescence. So we can yeah we can analyze, we can verify the cells that's under microscope. I mean fluorescence microscope or confocal microscope. So we can see, we can see the cell that yeah that contain that express that protein. So in this case, we use CRISPR-Cas canine complex, and we have canine that fuse. I mean the fusion canine that link with DFP. So uh, so actually the canine have all functions. So they can bind to the sequence of interest. They can bind to the RNA, and they can recognize the DNA, but they cannot cut the DNA. But uh, it contains the protein. I mean, not a gene, but protein. In the case, we have ZP, and so we know a specific we know the specific reason to locate the gene of interest, and to know, for example, we know that in that some certain gene have mutation or have no mutation, for example. Yeah, that's the purpose of this study. But this one is not very common, but we can do that. And the more common way is indel knockout, and we are homologous direct repair for gene editing. And so we have mutation, we have deletion. I mean, uh, we we miss one here. That is DNA. We have another application for CRISPR-Cas that have mutation in two different domain of the Cas9. So it's only cut the single strand and left another strand alone. So it have it's an application to delete. I mean to delete multiple side, multiple uh, gene sequence. Yeah, but so this is the old version. We don't have it one, but if you have a time, you can you can search about about nick case, canine nick case. So that is type mutation. So it can recognize different side of I mean multiple side of sequence of interest, gene of interest. So here I would like to talk very quickly about using CRISPR-Cas9 in the animal. So firstly, we determine the sequence of interest, and then we design the RNA, CRISPR RNA, and then we infect that complex. I mean, so for different purpose, for example, we inject that too. Normally, we use plasmid, but normally in animal cell we don't use plasmid. Yeah, we can use like two parts RNA and to insert into the cell. That is the example for uh, for generating animal model. So here we have, for example, in this case we have two RNA that can interact to, for example, in this case we have nick 2.5 gene, and we have another RNA that target to 640 gene. Yeah, and after that we insert that two RNA with canine complex into the embryo. So normally we have to insert the complex, I mean the CRISPR-Cas complex into the embryo day zero, I mean, as the state of single cell, single embryo. So, do you have any ideas for a single embryo why we need to inject a single embryo state, but not for like later state of embryo development? Any ideas?
Maybe because we need it to be modified before it differentiates in other part of embryo.
Right. Any other idea? Nguyễn Văn Anh.
I'm here. I think maybe because the embryo cannot inject, it cannot reject the CRISPR-Cas9. I don't know. Can you hear me?
Yes.
Yeah, I think it's easier to manipulate the genome in embryo state.
I need more idea. Hà Nhung. Why you need to inject the CRISPR-Cas complex into the embryo as a single cell embryo state? Sorry. Why we don't? Yeah. Why don't we inject the CRISPR-Cas at later state of embryo development, like as two cell, 3 cell, 5 cell embryo? Anyone? Any ideas?
Yes, I have an idea. If you inject CRISPR in only one in the single cell embryo, then the success rate is very higher. Like if we inject CRISPR-Cas9 in single cell embryo, then it can replicate along with the replication of the embryo.
Any ideas? Yeah, I see the message online. So can I explain for everyone? Can you talk a little bit more for everyone?
So basically from the single cell embryo, so what do you mean single cell embryo? It means you have one cell, and after that embryo will be will be divided into more cell, right? So if you do it on the first cell, then the genome will have the content in the editing, and after they divide into multiple cells, all those other cell will contain the editing, and you don't have to do it again and again and again. You just only need to do it in one cell, and that all of the other cell will contain the editing.
Right.
That's all.
Right. So that is perfect. Yeah, answer. So Yeah. So that reason. So actually, so that's the reason we need to do at early state of embryo. Yeah. Um. So here's some application in human. I mean, in disease treatment. But as I mentioned before, we still in developing, still in development. So we still have some issue that need to improve. But the most, I think the most positive is uh, to focus on some on cancer treatment and some kind of cancer treatment. Actually, some um, blood cancer because so normally we will not focus in blood cancer treatment. We will not focus on the cancer cell, but we focus on the human immune cell, especially in CD4 and CD8 cell, so T cell. So normally CD4 cell and CD8 cell, so they are both T cell, but we're more interested in CD8 cell. So the the thing is we want to introduce some normally we want to introduce some gene into the cell, and after that the cell can recognize, yeah, can recognize the cancer cell. I mean, T cell can recognize the cancer cell and kill the cell. So that's the most positive that we can do recently. And actually we have some cell therapy that based on not CRISPR-Cas, but based on gene editing. Actually, gene editing, but not actually CRISPR-Cas. So I think that is the most effective for cancer treatment. But uh for like to, I mean to CRISPR-Cas to edit the cancer cell. I mean the cancer in human body, we still have so many issues. Yeah, because we cannot inject CRISPR-Cas into directly into the cancer cell and to defunction some gene. Yeah, because we still have a lot of problems. We can do in cell line, but for the cancer cell it will be different um, because we have to find a way how we can deliver the CRISPR-Cas into the cancer cell, because cancer cell is stay in the tumor microenvironment that contain different things. Contain a cancer cell, it contains stromal cell, I mean the normal cell. It contains fibroblasts and different human immune cell for T cell, B cell, and other blood cell. I mean, so many things. So that's a problem, not only for CRISPR-Cas technology also, but it also for drug deliver. I mean, for chemotherapy that we want to use a target drug to, I mean, to a specific drug or um, some antibody to target specific cancer cell, because the the drug cannot, yeah, we have difficult way to deliver the drug into the right cancer cell, because of tumor microenvironment. Yeah, and so in some case, not some case, but all the case, the drug or the CRISPR-Cas also not target only in treatment. It not only targets the cancer cell, but it also targets the normal cell. So that's the issue. We don't want to target the normal cell. So the normal yeah yeah. So um so that is some limitation for for the using CRISPR-Cas for therapy. Actually, we still have some, but we still have some limitation. Okay. I think before going to another part, I think I would like to mention about I think therapy. I mean, for genome editing therapy, I think if anyone have any idea about cell therapy or immunotherapy or anything, not CRISPR-Cas9 because I want to talk outside CRISPR-Cas. We talk enough about CRISPR-Cas. So anyone? Quân Anh, for do you know any technology about cell therapy, immunotherapy, or anything else that is very modern technique, modern. Quân Anh,
Modern technique.
yeah,
I don't work with self and genes that much, so
right. Anyone have any ideas? Anyone? Yeah. Anyone who about Cas9? No, not Cas9, but CAR T cell, CAR T cell, CAR T. Okay, so I think the most effective for cancer treatment right now is have very best uh, response, very good outcome for cancer treatment is using uh, CAR-T. One of most effective that I know for cell therapy. So so normally in in our so in our body, I mean in human body or even in animal, so the cancer cell become the cancer cell develop from the normal cell. Right. Exactly, Quân Anh. Chimeric antigen receptor. Yeah, T cell. So we call CAR T cell. So normally, because the the cancer cell and the normal cell share the same genome, share the same morphology, everything quite similar. But the cancer cells that have some mutation, it accumulates mutation during their lifetime, and so it develop, it convert, it can transform from the normal cell into abnormal cell, and after that it acquires more mutation and it develop into the cancer cell. But in general, it shares many different similar. I mean, many common, not different, many common similarities. So therefore, the T cell cannot normally it cannot recognize the cancer cell. It cannot know that that is a bad guy that need to kill. So therefore, yeah, the tumor cell, I mean the cancer cell can escape the immune system and to develop, and to multiply. So so recently it's very, I mean uh is that few years, so chimeric CAR T cell. Okay. Chimeric CAR T cell were approved by FDA in 2020 to treat uh, lymphoma and also to treat different, I mean, to treat different kind of lymphoma like diffuse large B cell lymphoma. So we that engineer us, I mean engineer the cell, the T cell. So we use the lentivirus to introduce the gene of interest. Yeah. So actually to introduce the gene that express a specific receptor. I mean that a chimeric receptor that can recognize some protein. For example, in K for lymphoma, we have a CD19. We have CD19 antigen. So that expressed in all kind of B cell. but lymphoma is the cancer from B cell. So therefore, the T cell can recognize the cancer cell and they kill all the cancer cell. So that we call the tumoric antigen receptor. So we call CAR T cell. So now we have lymphoma CAR T cell and we have myeloma CAR T cell. So we have this one, myeloma CAR T cell and lymphoma CAR T cell. It works very good and it can kill some patients who have no way to treat because normally lymphoma can be lymphoma patient can be treated by different drug. I mean, can treat by chemotherapy, but when they left with chemotherapy, they cannot treat with any other drug, so they have no way to survive. So that's why we need another method and another technology, and so now we have CAR T cell. So if you interested about CAR T cell, you can read about this and you can see that it's very interesting and very helpful. But we still have some problem with CAR T cell because it works very good with myeloma and lymphoma cell, but it cannot work with tumor cell because tumor cell have a microenvironment that cannot allow the T cell, the T cell that go easily into the tumor core to recognize the receptor and also recognize the cancer cell. So so that's why we're still in development for the solid cancer. Okay, so for experiments, so I think I can let Dr. Ming to help you to design to learn it. Is that okay for you for next lecture? It takes like 30 minutes to complete this one. It's okay for you guys, everyone.
It's okay.
Yeah, it's okay.
Okay. Yeah. Because I have something. I have need to do something for my professor now. So okay. I think before finishing, I would like to have some summary. Yeah. Anyone can summarize this. I mean, lecture today. The today lecture. Quân Anh and Vân Anh, right? Can you summarize, some key points that we learned today, like how CRISPR-Cas work, how they recognize, how they prevent the bacteria. I mean, they help bacteria prevent virus infection. Yeah. Maybe Quân Anh go first. Is that okay for you?
Okay, you want me to talk about the summary of CRISPR-Cas9 from the first part of our lecture?
Right.
So for CRISPR-Cas, it have three components of this, including the CRISP RNA, the track RNA, and the Cas protein. So in the first step is the adaptation. The bacteriophage, when bacteriophage enter the bacteria with the viral DNA, a part of the viral DNA will be guide to with Cas1 and Cas2 to into the CRISPR array and create the and into the CRISPR array as part of the CRISPR, and after that, for the next step, the CRISPR that they will be transcribed into the CRISPR RNA, and this CRISPR RNA will bind with the track RNA and to create a duplex. And this duplex, after maturation, this duplex can combine with the Cas protein into the Chris RNA track RNA Cas protein complex, and this complex can when another time that when the bacteriophage send the viral DNA again, then this complex can recognize that viral DNA and start to degrade this viral DNA and protect the bacteria from that virus. Is that correct?
Mm-hmm. Yeah. So you know, yeah, it's short, but it's quite enough. Phương Anh, Vân Anh, is that Vân Anh? Do you have any addition?
Uh, oh, I think Vân Anh has sum up the overall information. Well, I would like to add some information of the structure of the Cas9 protein.
Right.
So, apart from the guide DNA that Agone has explained to you, another important component of this CRISPR-Cas9 complex is the Cas9 protein, and the bacteria has the Cas9 protein itself. So it's really amazing. And there are three domains that need to be concerned. Is first of all is the H and H domain and the RUBC domain. They are two domains that work together to break down the viral DNA.
Right. That one. Yeah.
Yes. And
that one. Yeah.
Yes. And the another important domain is the palm interacting.
Right.
It will recognize the palm motif on the viral DNA so that the Cas9 can bind to the viral DNA and cut down.
Right.
So that's all I want to say. Thank you.
Right. So, in general, I mean, in vivo or in vitro, I mean, in the cell or in vivo, for example, even you can do CRISPR-Cas. I mean, you can digest the DNA in vivo condition. I mean, without cell, is that need only CRISPR structure, Cas9 and guanine. You can dilate. You in the tube. It can also digest the sequence of interest. So in both case, in the cell or inside the cell or outside the cell, the CRISPR-Cas system can work similar way. Yeah, and the way to, I mean, we have two different ways for CRISPR-Cas. We have homologous direct repair, and we also have a non-homologous and repair. In two ways, it occurs randomly. But it's based on the DNA direction. Either we have a overhang or we have blunt DNA, so it can go to different way. But sometimes it can make like we cannot distinguish in some case between the non-homologous in which pathway it goes to direct into homologous direct repair, or in which pathway, in which case it goes to homologous direct repair. But normally for homologous direct repair is more common in gene editing that we use to edit to introduce the donor DNA into and to insert or to replace the some sequence of interest. So that is for for that is the highlight for homologous direct repair. And for non-homologous direct repair, we use for knockout or knock kill. Yeah, no, most in most case it will be knockout. Yeah, because it's very easy. Is that change the frame shift, insert or then list some nucleotide so we can train the whole sequence, whole frame, whole open reading frame after that mutation point. Yeah. Yeah. Okay. That is another highlight. And another highlight for in so in I mean in nature it will so in nature we have two parts guiana. Yeah. We have two parts guiana and we have a universal tracer RNA that can recognize yeah almost every keeper RNA sequence by constant reason. Yeah, normally. And but in engineering, I mean in research, we have two different kind of RNA. We have two-part RNA ELISAs, or we can have a single RNA. Single RNA. Single RNA. So here, so in research we have single RNA like this. So because we want to combine all tracer RNA and RNA and keeper RNA into the same RNA. Yeah, same RNA sequence. So we have single RNA. Yeah, it contains everything. But each one have its own benefit and difficulty. Yeah. For this one, it's easy to contract, so you can use like only one plasmid to contract the RNA, but you have some I mean difficulty in technique like how to insert the plasmid into the cell and how to to control the efficiency. For example, some cell like it's very difficult to inject the foreign DNA into the cell into their I mean pass to their membrane. So for but on the but for this one for two-part RNA, so we can insert into the cell by electroporation. So we use a machine with it can generate very high voltage, so it can break down the membrane and it makes the hole on the membrane so the RNA can easily go inside the cell. So it have very high efficiency in the in some kind of cell that very hard to transfer. So each one have its own benefits and in which case we use two-part RNA, in which case we use single RNA. It depends on the purposes. Okay. So I think we any question? Any question? Vân Anh, no?
No, I don't have question.
Okay. So so I think we can stop here, and so I would like to thank Vân Anh, and someone I cannot remember all of you, but I think someone have a very good contribution in this lecture today, especially Vân Anh and Quang Anh. I think Vân Anh know very well about CRISPR-Cas, and Quang Anh know. Yeah, I think Quang Anh very. Yeah, I think Quang Anh very quickly. He I don't know, but I would like to thank to